You know, thanks.
Okay. So I'm delighted to be able to speak to you today. This talk is a little bit different than what's been on the workshop so far, but I just thought I'd try and provide you a perspective of our use of sampling algorithms in modeling biochemical networks and what suggestions we have from that field for continued development on the theoretical and computational side that might interest some of the members of the audience. So first I'll introduce you to constraint-based modeling of biochemical networks. Then we'll discuss applications of uniform sampling of polyhedral convex models. And then some non-uniform sampling of also polyhedral convex models. Some efforts in our field to sample non-convex feasible sets, and discuss a bit about entropy optimization and its relationship to constraint-based modeling and how that relates to a problem concerning sampling intersection of a polyhedral convex set and a convex cone. So just a brief thing on notation. It's all pretty much household notation. It's fairly standard. The only thing that sometimes people would notice is if I say log x, for example, is the component-wise logarithm of each element x. So in biochemistry, you can consider, or in modeling in general, you can consider two broad classes of modeling. So what I would call generic modeling is mathematical modeling approaches that don't satisfy any mechanistic modeling principles a priori. So, for example, if you were trying to infer the network of a biochemical system, you might have no restrictions on the class of topology of the network you were going to infer. So, no restrictions that arise from, for example, mechanistic considerations like physical chemical laws or biochemical principles. Mechanistic modeling, in contrast, you would have mathematical model that includes specification of assumptions that require certain mechanistic principles to be satisfied. So, for example, in a mass flow network in biology, you would like to have all the networks to satisfy mass conservation. So that's a principle that you can convert into a mathematical formalism and it aids you in the modeling. But these are sort of two broad classes, and there's of course mixtures between the two. But within the mechanistic modeling approaches that are applied to biology, there's two broad classes then, and the first one is differential equation based modeling, and typically here you'd have your input would be your biochemical network and your some initial conditions and parameters if you had them, and then the output is some unique temporal trajectory that displays the state of your system over time. In parallel with this, more common in biochemical network modeling by now is what's called constraint-based modeling. And so here your input, again, you have the network topology, but you accept the fact that you don't have a complete knowledge of the initial conditions or complete knowledge of the parameters, both of which are the result of millions of years of evolution and to some extent are not known, and other people would say are not knowable. So you accept that the output, or in other words, the set of predictions you make is a set of is no longer unique temporal trajectory, but some sort of set of temporal trajectories, or it might even be a set of trajectories that are invariant with respect to time, but it's not like a unique prediction as such. So the fundamental unit that we are trying to model here in our system, let's say one link in the larger network, is what's called a chemical reaction. And here you have substrates indicated in blue and red, in this case hydrogen and oxygen, and they're converted into products which are in this case water. And this reaction, it transforms hydrogen and oxygen into water. So all these reactions are in principle reversible. In practice, sometimes only one direction is present. So if you're thinking about this like a road network, sometimes you can only go one way down the road. To model this dynamically, what's typically done is set up a system of dynamical equations where you're assuming that the probability of a reaction is proportional to the concentrations indicated by C here of the different metabolites involved. And it's actually interesting yesterday listening to the talks in the rest of this workshop on how much of the theory is related to the underlying considerations in statistical physics about flows and for example, mentions of detail, balance, etc. Those are concepts originally came out of physics, and eventually, if you track them back, you can track back the same concepts being related to these equations, which I'm showing you here in terms of the probability of mass flow in biochemical networks. So, unlike many physical systems, however, detail balance doesn't hold in biochemical networks because there's constant input and constant output. So there's always a net flow in one direction. You can set up a system of equations like this, these elementary reaction kinetic equations, and the coefficients there, which you can see in the top line, the 2's and the ones that are not apparent, they can be amalgamated together into what's called a stoichiometric matrix. And so then you get your dynamical equation with respect to time is a product of this matrix times some function of the concentrations. So that's the typical dynamical equation-based approach to modeling biochemical networks. The advantage of this, of course, is it's chemically correct, and the nonlinear relationship between species concentration and rates is properly represented. You can do steady state and dynamic modeling, and you compare with different types of data. It's necessary to do this type of modeling for certain types of biochemical processes. And there's a rich variety of mathematical and computational tools, as you know, for modeling with differential equations. However, there are plenty of disadvantages to this approach. And so the main one, as I mentioned earlier, is the initial condition. You often don't know it. So you don't know the initial condition of the system in detail. So it's not like a cannonball fired out of a cannon where you know the position of the cannon ball at the beginning. You don't know most of that sort of information for biochemical systems. And also, you don't know the kinetic parameters. So it's typically like you don't know the angle of the cannon, exactly which way it's pointing. All you know is the cannon is not pointing directly towards your own army, but it's kind of pointing off to the distance at the other army, but you don't know exactly where. So that's the typical analogy one would give for the knowledge of biochemical kinetic parameters. So given those constraints or difficulties, more perhaps fundamentally is the criteria for convergence of the system to a non-equilibrium steady state are not known. So there are plenty of theorems for convergence towards what would be called an equilibrium steady state, which is equivalent to detail balance. But as I said, detail balance doesn't hold, and so we're in a non-equilibrium steady state. But actually the criteria for the dynamical system to converge to such a state is not known. And it's also difficult to fit the parameters then, even if you're given plenty of dynamic data on the concentrations. So there's plenty of approaches to try to reformulate this thing, but all of them end up with the same type of problems, which is having local minima, can't fit the parameters, aren't enough parameters, etc. Initial conditions not known. So it's currently intractable to take this approach for modeling high-dimensional biochemical networks. In contrast, constraint-based modeling is suitable for modeling at genome scale. And the principle here is to eliminate infeasible network states with mathematical specified constraints. So for example, mass conservation will be turned into a set of constraints, and then you apply those as to eliminate possible network states that are incompatible with known physical, chemical, or biochemical principles. So, for example, a biochemical constraint might be that vitamin C is essential for a human biochemical network, but not for a mouse biochemical network. The challenge here is that there are not enough constraints to uniquely specify the dynamics of the system, and you can't specify them in detail enough. There isn't enough information to specify them. For the same reason, the dynamical equation modeling is difficult. And so you get non-unique predictions. So for example, if we assume mass conservation in steady state, so in other words, that internal rate of production of each metabolite and external input of each metabolite is equal to the consumption and external output, we get a steady state. This steady state assumption then can be just represented as a matrix vector product between the stoichiometric matrix and a flux vector. So the flux vector is like the rate of flow, or the rate at which each reaction occurs in the system. So the analogy for a traffic network might be the net traffic flow between two cities or something like this. So this constraint then has been applied in what would be called a prototypical constraint-based modeling approach, which is to set up a linear optimization problem where your objective is perhaps to maximize the growth of the organism subject to steady state mass conservation constraints and some bounds on the uptake and secretion of particular metabolites. So those bounds are L and V indicated here, or L and U indicated here, and V is the rate of the reaction. So this problem here is, of course, it's a straightforward linear optimization problem, and it does have particular features, but for the moment you can just consider it like a straightforward linear optimization problem. This is the most common approach to genome scale biochemical network modeling. The advantages of it, of course, you can just reuse plenty of algorithms that are already out there. It's suitable for high dimensional modeling, and it's accessible to a broad user base because it's not so complicated to understand linear optimization, even if your background is in biology. Disadvantage here is you have to hypothesize some objective. So what happens when you don't actually know the objective of your system? So, for example, a typical scenario would be, let's say, if you're trying to model the growth of a bacteria in a rich nutrient media. Well, a reasonable approximation in that case is to assume that the bacteria will try to replicate as fast as possible, and so you have a decent idea what your objective D might be. But the objective, for example, for neuronal metabolism or liver metabolism, etc., it's not to grow as much as possible. That's would be equivalent of liver cancer or something like this. So we don't know what the objective is, and so in those sort of scenarios, then we come to the scenario of sampling the set of steady state solutions. So this is now the start of the connection between biochemical network modeling and sampling algorithms. So there are many applications of this type of approach. And I'll just only mention some examples. So, for example, you can use this for design of experiments to predict the most informative measurement to make. If you can measure some of the L and U's, you can constrain your space well. You can use sampling to predict which is the most important ones to measure to reduce the volume of the solution space, or the volume of the set omega there as much as possible. Volume computation is important. So it was shown at one stage that genetic defects that decrease the volume of the solution space were most likely to have a clinical effect in vivo. You can then interpret the marginal distributions, for example, to see which set of reactions are always required for a microorganism to grow in many different conditions. You can also use it as a way to detect which are the reactions that are most important to consider for changes in, for example, conditions like diabetes, etc. So these are only three examples, but there are many different types of things. There's hundreds of studies using sampling in biochemical network modeling. Recently, there was a review of different sampling algorithms available or actually used in biochemical network modeling, and also available as code. And this is independent. I wasn't involved in the study now, but the outcome of that study here is the Pullman, the Amolin, and Arnold are three different types of models, and they're just showing you performance of the 3 different sampling algorithms here. And the outcome was that the CHR, the coordinate hit and run with rounding, what was the most most efficient and highest quality algorithm that was used in biochemical network modeling sampling at the time. So the plots on the lower left, they indicate the trace of, you know, each time they're looking at one of the variables to see is it correlating with each other, and the second one is the autocorrelation. So you can see the autocorrelation for the coordinate hit and run with rounding is near 0, whereas for the optimized general parallel solver and the artificial centered hit and run, it's not, the autocorrelation is substantial. So the coordinate hit and run with rounding, you got some talk, a very nice talk yesterday from Aditi on that, and I guess tomorrow Professor Van Pala will also mention it. This is the one that we first integrated with the COBRA Toolbox ourselves, although the artificial centered hit and run was already there previously. And I had long-standing worries that we had no guarantee that the artificial centered hit and run was going to be sampling uniformly. And so we were using it on large scale models, but we had no idea that it was really going to be sampled uniformly. So it was nice to get the reassurance of having an algorithm in there that had strong theoretical foundations, because you can be making important conclusions based on sampling, and you want to have reliable, using reliable samplers. So there are various challenges still, some of which are being partially addressed by existing samplers that are in use, but that I would like to introduce to you as we go along now. And I say one of the main ones I think that are really will require substantial effort in your field will be to dealing with anisotropy. So, for example, in biochemistry, there is intrinsic multiscale problems there. So, for example, the rate of reactions in metabolism. So that would be, let's say, the rate at which you uptake glucose from your gut. That rate is far faster than the rate at which you, for example, convert a messenger RNA into a protein. So different levels of biological organization have intrinsically different levels of rates, but those two systems are completely interdependent to each other. You can't split them apart. So when you're trying to model the whole, in other words, for example, metabolism and macromolecular synthesis or expression, when you're trying to model those two together, you end up with these linear models that are multiscale, and they create very anisotropic solution spaces or feasible sets, in other words. An example of a stoichiometric matrix here is provided, and this is one of the first integrated metabolic and macromolecular expression models. So typically the dimension of those is in the tens of thousands. The coefficients are very sparse, but individually they're spread over 5 order of magnitude, and the color bar here is indicating the number of standard deviations away from the mean. So there are some very small coefficients in yellow and then some very large coefficients in black, and they'll be spread over many orders of magnitude. Not shown here, but just to say, is that the bounds, the L, the lower bound and the upper bound on the variables are also spread over multiple orders of magnitude. So it's not a trivial problem to bring these feasible sets into isotropic position. Even in linear optimization, these problems can kill the existing industrial optimization solvers. For example, Garobi, Cplex, etc. They will either report that the problem is infeasible or solve it, and then after rescaling the problem, the solution will not be satisfy the original equation. So together with Michael Saunders and others in Stanford University, we developed a quad precision routine for solving these linear optimization problems. So you could start the problem in in a double precision LP solver, then you use that to warm start a quad precision LP solver, and then you rescale, you take away the scaling that you applied, and then finish it off with a final quad LP step, which then gives you your quadruple precision solution to the LP problem. So the reason I mention that here, even though we're talking about sampling, is this is the sort of thing that will be necessary, I think, or some variant thereof, within the sampling community if you really want to tackle the multiscale nature problems in biology. Another big challenge is dimensionality. So you can see here, as we go from 1995 projected to 2025, the model sizes are going to be in the hundreds of millions. And so we're already sampling models of about 5,000 dimensions, and Professor Van Pala will hopefully indicate tomorrow the progress on larger models in biology. But suffice to say is that that's not going to be enough. You know, if we want to be still doing this sort of thing in 10 years' time, we really need focus on very efficient algorithms and scalable algorithms. The other thing that's really quite important for us in practice is knowing the convergence criteria. So, you know, okay, it's great to know that it's convergent to a stationary distribution, but like, is that uniform? Because if you ask for a uniform distribution, is it really uniform by now? Or how close to uniform is it? So I see a lot of the complexity analysis this week has been about, you know, how long does it take to compute the samples, et cetera, But in practice, actually, more the issue for us is like, well, how many samples is enough, you know, and defining what enough is also, it can be tricky. And so what you can see here is, depending on the number of steps in the CHRR algorithm, we get either a unimodal or not distributions. And of course, this should be a unimodal. But knowing for various algorithms how this, how much sampling is enough is an important challenge. So we have disseminated together with collaborators I mentioned already the algorithms for sampling through what's called the COBRA Toolbox. It's a MATLAB suite for prediction of modeling with constraint-based modeling of biochemical networks. It has it's very widely cited. The website is visited 1,000 times a month, about 120 Git clones a month, and it's the largest collection of constraint-based modeling algorithms. And so in there we have now of late recently added log-concave sampling of polyconvex sets. So we've mainly been talking about uniform sampling so far, and of course for any biologists in the audience. So there are the log-concave function you can think of. It's like a unimodal function. So an example of that would be a normal distribution. So an example of using this in biology would be if you had information not just of the lower and upper bounds for your reaction rate V, but if you had experimental data that was the mean of some measurement of the rate, it typically comes with a standard deviation, and then you can use that mean and standard deviation as input data now to the sampling algorithms, and then you can sample from a normal distribution over the polyhedral convex set. So this approach has been implemented by Ruqi Shen, Yintat, and Santosh Van Pala here, and it's integrated already with the COBRA Toolbox. I guess Professor Van Pala will talk about that more tomorrow. And this is just an example showing you here, when we use this sampling algorithm, we can compare for reactions that weren't constrained with such experimental data, what the predicted sample distribution in pink is, and compare it with and it compares favorably with the experimental mean indicated in yellow. So the blue there is just an alternative approach to just predict a single flux vector using two-norm minimization. This sampling algorithm is interfaced as a submodule within the COBRA Toolbox, and that would be an approach we would advocate for other people in the audience that would be interested to contribute their sampling algorithms. As you maintain your own Git repository, but we include it as a submodule and work together to make sure that it can access via a common interface. So here, currently in the COBRA Toolbox, you can use the CHRR or artificial centered hidden run or the Riemannian Monte Carlo algorithm via a single interface. And this is also might be quite useful for you if you want to compare different sampling algorithms and how they perform. Because just changing one line of code, you can switch from one algorithm to the other. A challenge here really is, is having findable, accessible, interoperable, and reusable software. So, a lot of the software that's built is not satisfied as principles. It tends to be just developed by a single PhD student, and they don't seem to think about the investment in time and effort that's gone into their PhD, and that loads of people might really want to use their software, but they can't because it's not accessible to them. And so I think if there's more focus on that sort of thing in your community, in the sampling community, there would be increased impact of the sampling work, and this would facilitate arguments for greater investment in fundamental and applied research in this area. So it's a win-win situation from my perspective. Moving on now to other types of applications. So another disadvantage of flux balance analysis is that important constraints are missing. So this linear optimization model, it doesn't include all the constraints we know are important. And so this has motivated efforts by people to try and incorporate further constraints. So for example, if you consider a biochemical network as a linear resistive network where you're given a set of resistances, you can set up a quadratic optimization problem, and the optimal conditions to that, when combined together, will satisfy Kirchhoff's voltage and current and voltage laws, which are equivalent to energy conservation and mass conservation in this case. And then Ohm's law is relating the change in potential to the rate of the reaction. But this constraint and the optimality conditions, it only specifies the flows and the change in potential if you're given the vector resistances, but we don't know the vector resistances. So people have been trying, in my community, have been trying to sample the non-convex set of optimal solutions. So I'm talking about the full set of solutions if you consider R a parameter. So if you were to sample R, what set of different optimal flux vectors would you get? So it's this set defined here, and there is a bunch of papers now in my community who've been developing algorithms for sampling these sets. So what my question is what is the relationship between these algorithms and the theoretical and computational literature in the computer science community? So these are some of the papers here. Some of them might have interesting ideas for you, and some of them, they might anyway. I think they deserve more attention, and perhaps criticism. More generally, going away from just pure quadratic optimization, we consider that every constraint-based modeling problem can be formulated as an optimization problem. So even if we get an optimization problem with a unique solution, invariably that optimization problem will be have an input, some parameter, and the parameter itself will be unknown. So you might want to set it up, each optimization problem with a unique solution, but actually, even if you do that, you have an input parameter that's somewhat unknown, and you want to be able to sample the set of optimal solutions as a function of the input parameter. And so the challenge I would put forward to the audience there now is, how do the established sampling algorithms map onto the sets of solutions to different classes of optimization problems? Is there solutions, are there algorithms out there we can already be using? And if not, why could some of those be developed? Because they would be very useful. And along this vein, if you consider a non-linear resistive model now for a biochemical network, where you're maximizing the entropy of a pair of unidirectional fluxes. So the net flux being the difference between the forward VF and the reverse VR flux. So maximizing the entropy in this case will at the optimum will give you energy conservation and the second law of thermodynamics holding at an optimal solution. And there's a nice interpretation of this because it's the least biased prediction given the data. However, there's still a vector of free parameters. So the question I would have then is, how in principle would you sample the set of optimal solutions to this type of problem? I've been thinking about it a little, and this is as far as I've got. So if you consider this general entropy maximization type problem, you can reformulate it as a linear maximization problem subject to an intersection of a set of linear and exponential cone constraints. And so the concept I had was that if you could reformulate the nonlinear terms in your objective into a set of convex constraints, could we then somehow sample this more complicated intersection of constraints because it's still convex, and in that way would it be possible to sample the set of optimal solutions to this problem? Reason why that would be interesting then is if you took the unidirectional fluxes and took one from the other, you would actually have a solution to this type of problem here, where we know the solution set is non-convex. So there's a relationship, of course, between those two spaces. So to summarize, I just like to highlight to you, I hope I have tried to highlight to you that constraint-based modeling of biochemical networks, it provides a strong demand for sampling algorithms and a host of challenges. The intrinsic anisotropy I've mentioned, the high dimensionality and increasing dimensionality of these problems, the need for practical convergence criteria and thinking about the computational costs of checking whether something's converged to the stationary distribution, but also the distribution that you are actually wanting the algorithm to converge towards. Because, for example, if someone was to make a mistake in the code, it might be converging to a stationary distribution, but is it the stationary distribution that was desired? These sort of things, they need to be checked. The software that's developed, that needs to be accessible to others. And I've provided you an example of a route how we can maybe help your community make the software accessible to biologists. There needs to be hopefully more communication between the computer science and the mathematical optimization community, because I think in parts they're running in parallel at the moment, and they could be learning more from each other. And then this sort of more general question I have about considering feasible sampling problems and parametric solution sets of optimization problems, like what the relationships are between those. So I'd like to acknowledge the collaboration with Professor Pampala and his students Ben and Aditi, and also with Yin Tat and University of Washington, Professor Ines Thiele in Luxembourg, Michael Saunders in Stanford, and my students and postdocs Hulda, Herman, and Herman. So thank you for your attention.
Thank you very much for inspiring talk. Are there any questions? Okay, I need to bring the microphone over.
Thank you very much for the nice talk. Wonderful applications. I'm curious for the matrix, like the 60,000 matrix, how do people usually specify those entries? Is like every entry correspond to some research paper or like well-known scientific fact, or like so you have very small set of simple principles to determining those entries?
Both. So typically, so there are different types of biochemical reactions, right? So in many cases, in what's part, let's say, metabolism, one PhD student is responsible for one row of that constraint matrix, or one column, actually more like it, one column. you know. And so that they never anticipated that their work would be abstracted and put in a computational model many years later, but that's actually what happens. So then you get PhD students that collect this literature information, abstract it, and manually construct these matrices. Then you get other parts that are also manually constructed, but they're more like template reactions. So for example, the set of reactions to convert a messenger RNA, like what you get in the Pfizer vaccine, into a protein. The set of reactions there, the reason why that vaccine method can work is there are a set of template reactions in your body that will respond to whatever messenger comes in. And so when the messenger comes in the vaccine, it generates the protein using a set of these template reactions. And so we can specify part of that stoichiometric matrix by using a set of template reactions, and then we just input the set of genes that are in that organism. You know, so it's a mixture between fully specified and reactions, like let's say in terms of manual specification, and sort of algorithmically specified ones.
Hi. First of all, thank you for. Oh, is it on?
Yeah, it's on.
Oh, okay, great. Thank you for the talk. Yeah, it makes me really happy to see this kind of talk also as part of the conference, and I fully agree with your sort of call for more interdisciplinary communication. And I have a question that is not directly related to the work that you presented on sampling, but rather on what you explained at the beginning about the choice of formalism of how to represent these processes. So something that I came across recently is these hypergraphs for chemical reaction networks. I don't know if you have heard of them. I'm wondering if this is, you know, more of a mathematical curiosity, or if this is something that's actually used by people. If so, the idea is to have different chemicals represented by nodes, and then to have an hyper edge, which is not just an edge between two nodes, but going from a set of nodes to a set of nodes that encode chemical reactions, and then to use this graph structure on top of it. I don't know if you have any experience on that.
You just happen to be talking to the right person.
Okay.
So we recently published a paper that establishes that biochemical networks are not any arbitrary hypergraph. The hypergraph formalism is very useful because it makes people think that they're straight away they're not graphs, right? Because even in the example I showed at the beginning of the hydrogen and oxygen converting into hydrogen peroxide, so that reaction is on one side there's two nodes and on the other side there's one node, so right away it's a hyperedge. Okay, yes. So it's great that people start thinking about hypergraphs in biology, right? On the other hand, it's actually not so good, because as you know, there are so few theoretical results for hypergraphs, whereas there's lots and lots of results for graphs, like mathematical results, right? So actually, biochemical networks are a very special type of hypergraph. And if you take the incidence matrix for a biochemical network, the stoichiometric matrix that I showed you there, it can be split into the sum of a set of incidence matrices, each of which corresponds to a graph.
Yes, you can always reduce it to normal graph, right?
With certain exceptions, right? So actually now we don't know, is this always true in a topological sense? But it seems to me that there's a special type of matrix, that stoichiometric matrix is special. You know, it's not an arbitrary rectangular matrix. That's definitely the case. But it can be split into the sum of a set of matrices that correspond to graphs with some coupling. So the way to think about it is it's like there is each biochemical network is a hypergraph, but underlying it there's a set of graphs, and the flow in there are the invariant chemical units. you know. So, and then like inside that, then the flow of these invariant chemical units is sometimes hard coupled. So the rates of transfer is the closest analogy would be multi-commodity flow networks in transport problems. But in that case, let's say if you have a ship or a lorry with a bunch of oranges and apples, then the rate of flow of oranges and apples can go through the network at different rates. But those sort of constraints are hard in biology, in sometimes in the sense that it's exactly 2 oranges and 1 apple, you know, per unit time flow, you know. So I hope that ping me later and I'll send you the paper.
Very interesting. Thank you. I mean, I will look at your paper and then maybe get in touch. I'm happy to hear that. Thanks.
Yeah. I'll just follow up on that. The reason I mentioned it is I'm really trying to get linear optimization people to consider this not like an arbitrary rectangular matrix, because the network flow algorithms are so much faster. So.
Great. Thank you. Since I also appreciate you kind of trying to bridge this, make these bridges with the theoretical community and give us somehow feedback as to what is that you really need. I really actually appreciate that. Can I just ask one question? It was a little bit fast for me at the beginning because, again, you would be the person to ask. So once you get that linear program, why is that you wanted to sample the uniform measure on the polyhedron of the set where the constraints are satisfied?
Well, initially, because we had no better idea, right? So you're talking, I wasn't me now, but I mean, this was right back in the day when only the artificial centered hit and run algorithm was available. And the idea was, it's the opposite of hypothesizing a linear objective. You're not hypothesizing any objective at all. It's like it's almost the least biased type of thing you can do if you sample uniformly. Now, statistically, perhaps that's not really the case. Like, so some people would say maximizing the entropy subject to the available data is the least biased prediction you can make. But it started off as uniform because they were the algorithms that were accessible. It's not just the algorithms that are done theoretically, but the code that was available and understandable to use, you know, that often dictates what's used.
Mm-hmm. But what you're saying, basically, there might be, you know, some, you know, functional that dictates kind of what distribution you want to sample on that polyhedron, right? Which would also be something that kind of this community, you know, is exactly working on sampling, right?
Yeah, so all I can say to that is I'm super excited to start playing around with the Log-concave sampler over convex set, and there kind of remains to be seen how far we can push that. But an example I'll give you now is that the number of people in my field who would know what a Log-concave function is very small. And so you're dealing with a communication issue. So like I like to explain it to people as a unimodal function, even though maybe that's too broad or not exact of a definition, but it helps them say, oh yeah, I can see what you mean now, you know?
Thanks. Yeah, and you know, I appreciate this interaction. And okay, let's thank Ronan again. We continue at 11:30 then, right?